everybody to another Springpod session, this time with the focus on genomics. So we're very lucky to have Nick Hickson with us, who is the Education and Training Lead for the Southeast Genomic Laboratory Hub. And we have Layla Akfami, who is a clinical scientist and practice educator, who are going to do the talk with us today. So welcome to everybody, and I'll let Layla take on from here.
Great, thanks so much, Komi, and thank you those of you that have joined today and those of you that are watching on replay on YouTube. So as Komi kindly said, myself and Nick are here today to talk to you about genomics. This is the area we work in and are passionate about. So if you've got any questions, pop them in the chat. I'll try my best to answer them. So the outcomes today to learn or understand more about genomics as a career and learn about the different pathways into genomics. So we are based at the Southeast Genomic Medicine Service, but genomics is countrywide. So you do not need to be based in London to get into genomics. Firstly, what is genomics? I won't spend too long on this because I'm sure many of you know, but essentially it's studying all of a person's DNA. And sometimes there are typos in the DNA that cause disease. And our job in genomics is to find those typos and to explain what they mean for patients. So you might have heard of this analogy before, but I'm going to bring it back because I think it's a really fantastic way of thinking about it. Imagine trying to find a single spelling mistake in a book with 3 billion letters. And that is what genomics is like as a career. So you've got a book here, and a gene is thought of as a chapter in a book, and then you've got a genetic variant. So instead of the cat jumps, we've got the CGT jump. So the A has turned to a G, and we need to understand how does that change the meaning of that sentence, if at all. So that's our job. So first, I just want you guys to reflect and think about this. No need to answer, but what do you think a genomic scientist or someone working in genomics, what do you think they look like? I've got 3 images here. I've chosen them specifically for this. So just have a think to yourself for a couple of seconds. And if you saw through my bluff and the fact this is a bit of a trick question, you'll hopefully have said all of them. Because if I go on to the next slide, we've got lots of different roles in genomics. Obviously, the one that you think about first might be the one on the right-hand side. We've got a lady in a lab coat with a pipette, moving liquid from one tube to another. And that can be someone that works in the lab. So they do the hands-on science. So if you enjoy working in the lab and you enjoy using your hands in your work and things that are tactile, then you might enjoy this kind of role. So this includes working with samples such as blood and saliva. You might be running different kinds of machines such as next generation sequencing and doing all kinds of different experiments. Other kind of roles that we've got as scientists, so that's myself and Nick, we like to think of ourselves as the detectives in genomics. So we're mostly computer-based. We analyze DNA data and we try and figure out the answers for patients. So the image in the top is probably what me and Nick look like most days, scratching our chins thinking, how can we explain this genetic conundrum? And then we've also got people that work in admin. So we've got admin support roles, so that includes booking samples into systems, checking patient information, and coordinating between teams. Now, any of these roles can look like any of the images. So I say the top picture is what I look like most days, but actually occasionally you might need to go into the lab and do some lab work as a scientist. Or you might be doing a multidisciplinary team meeting where you're trying to figure out the answer with people from different teams, because it's not just a siloed area. Genetics impacts lots of different areas of healthcare. So you might look like the image in the bottom picture as well. So it's a really diverse area. And I wanted to see if you could have a go at figuring out one of these conundrums. So this is a little bit different, but bear with me. So put yourself into the shoes of a scientist. So you're a scientist for the day and data has been uploaded for a genetic test, which is done on the X chromosome. So I've analyzed this, or you've analyzed this, and you find one result to be slightly unusual. So what you do is you arrange a call with a senior scientist to discuss. Like I said, genomics is not siloed. We work with lots of different teams, including other people within genomics. So here we've got a call to discuss, to figure out why this result might be so unusual. So a bit of background, you might already know this, but the important thing to note with the X chromosome is that those that are born genetically female have 2 X chromosomes usually, and those that are born genetically male typically have an X and a Y chromosome. So we only expect to see 1 X chromosome in someone who's born genetically male, but we expect to see 2 X chromosomes on someone that's born genetically female. So that's a really important background knowledge to have before you try and figure out this conundrum. And this is the result. These are actual results, by the way. I've modified it a bit for privacy, but these are real results that we see in genetics. So this DNA trace is from a male patient's X chromosome. Now, we would expect to see a single peak as a genetically male individual, like I said, has only got 1 X chromosome, but this patient's got 2 peaks. Now, just to make it a bit more clear, I've circled the peaks that I'm talking about. So the bit at the beginning, they're called stutter peaks. We don't need to worry about those at the moment. The main thing are these 2 peaks. And also don't worry about the numbers either. These are sort of expansion sizes, which become relevant in other parts of genomics. But we've got 2 peaks. We only expect to see 1. This is a genetically male individual. You can either think about it yourself, or if you want, you can put your thoughts in the chat. I'll give maybe 30 seconds. Do you know how this could have happened? I'll give 30 seconds for you to have a think. Right, I think that we don't have anything in the chat, do we? Can I make no, that's fine. You might have been thinking about it, or you might not know the answer, which is absolutely fine. I just wanted to give an idea about what kind of jobs that we do. So here are 3 ways that this could have happened. We could have had something called mosaicism, which is where a person has 1 X chromosome, but 2 different populations of cells. So it appears as if there's 2 X chromosomes when there's only 1. So this person would still be genetically male. There's something called Klinefelter syndrome. So this is where a person has got 2 X chromosomes and a Y chromosome. Again, they appear male, but they've actually got an extra X, which is a little bit unusual. Or 3, it could even be a problem in laboratory processing. So sometimes people go into these areas and they think, oh, there must be a really elaborate answer as to why we've seen this weird result. But actually, sometimes there could just be a mix-up or another problem in the lab, and you go back and you get a repeat sample and it resolves and you get the answer that you would expect. So I put these into these slides just to show you the kind of things that you might come across working in genetics and the sort of problem solving and detective work that I previously mentioned. This is kind of what we are looking at. So skills that you need to be a good scientist, problem solving. I mean, we just saw in that example there, you need to be able to look at a problem and decide what are the potential answers and investigate each of those. Critical thinking, a lot of attention to detail. There's lots of small, tiny changes. And also, you know what, people don't often mention creativity, but I've thrown that in there because I think to be a good scientist or to work in something like genomics, you have to be able to think a little bit creatively and outside the box, because sometimes the answer that is there might not be what you first would have considered. And a bit about salary and progression. So NHS uses standardized pay bands. So if you've been to any of the other Springpod talks, you'll know about these. And the typical ranges are, so in administrative jobs, you might be looking at band 3 to 6, laboratory staff, band 3 to 7, scientists, band 6 to 8. So you've got from entry-level trainee scientists through to senior scientists. And you've got senior leaders, usually around band 9, very rarely sometimes, usually band 8, very rarely also band 9 for very, very senior leaders. And these are the pay brackets that I've rounded them, but they're correct at the time of speaking. So these could change year on year. So it's most of the bands that you need to be looking at, and then checking the agenda for change and seeing what kind of salaries they tie up to that year. And you can start at any level really, but usually people come in at entry level, so band 3 or 4, and then they work their way up to a more senior level. My colleague Nick, who's also on this call, will go through in much more detail the kind of entry routes for genomics later on in the presentation. So again, how do you get into genomics? Just surface level before I hand over to Nick. Apprenticeships, this is a relatively recent thing. It wasn't as big when I was getting into genomics, but I think they're fantastic. You get to learn while working. You don't have the problem of student debt that you might get at university. And there's lots of different levels you can enter as well for apprenticeships. Alternatively, university, I've just said the debt you get into, but actually that can be a really good entry point for different kind of people. That's how I got in. I did a degree in genetics, but again, you don't have to have done a degree in genetics to get into genetics. We get people with all sorts of degrees that come and work for us. There's also graduate schemes. So again, when I finished university, I then went into a graduate scheme called the NHS Scientist Training Programme. This is a rather competitive process, but it's not impossible. And it's a way, if you're really interested in becoming specifically a scientist, you can get into that through this STP route. Whereas if you're interested in working in a lab, you don't need to have done that. You can get in straight from university. You can get a job in the lab, or you can do an apprenticeship. So this is just to say there's no single path. There's loads of different ways in. So no matter what your background is, if you're interested in genomics, that's the most important thing. And I'm sure you'll be able to find the route that works best for you. And for more information, you can check the NHS Careers website and also the National School of Healthcare Science website. There's lots of really useful information there. So now I'm going to hand over to Nick. Nick, do you want me to give you slide sharing control? Oh, I can't do that. I'll just click through as
Yeah,
you
that's
as
fine.
you
Yeah, if you can click through. Thank you very much, Layla. So yeah, I'm just going to go into a bit more detail about the different career pathways within genomics. So this is not what I'm going to show is not necessarily representative of every lab in the country, but roughly it's very similar across the different laboratories in the country. So concentrating firstly on one of the pathways, which is our technical team staff. So as Layla mentioned, these are the people who do all the work in the laboratories, produce all the data before the data goes to the clinical scientists to do the analysis and the reporting, although some of the analysis is also done by the technical team. So starting the band 3 agenda for change, which is the standard banding that Layla mentioned, you could become a medical laboratory assistant, or MLA for short, and you can just get that job just by applying for it. As long as you fulfill the relevant person specification criteria. But you can also get into this through the apprenticeships, which, as Layla mentioned as well, is, at least in the southeast for genomics, it's a relatively new approach that we've been taking, and we've invested into these. So it's a really nice way of getting an entry-level post within genomics. You can get paid for working, you study through it, and you on your way to almost like a foundation degree. So it's the first step, level 4 apprenticeship. And then Layla's going to show now the next step, which is once you've completed that, you can move on to a band 4 or band 5 genetic technologist post. Again, this is something you can apply for directly. Usually there's a bit of a requirement. There's a requirement of potentially having a university degree for a band 5 post, although that's not strictly the case everywhere, but in the southeast it would be. However, you can also get in through the level 6 healthcare science apprenticeship, which actually leads to a university degree. So you're working for 3 or 4 years and getting paid, but you also at the same time get some study time to work through a university degree as well. So it's a really, really good approach, because you've got a job, you get a degree out of it in the end, and you don't have to pay for it either. And then once you've progressed, you can move on to the band 6 posts, which are the more senior GTs, so senior genetic technologists. And the next step is a band 7 post, which would be more of a bit more managerial post, so technical lab manager, and you could move all the way up to band 8, which tends to be one of the higher salary points for technical team members. But you know, if you look into it, there's band 8A, 8B, 8C, 8D. There's different pay scales there, but this shows you that there is a nice pathway for your career progression through the lab route, but this is also something that we in the Southeast, but also in other parts of the country, we're trying to create more and more roles that might give more options, in addition to what you can see here, to give more options for slightly different career pathways and career progression options, and to make it some roles that might be a bit more interesting for people who are not specifically interested in what you can see here. So it's in progress, but this is what currently exists. So the scientific team is the other career pathway. This has some more specific requirements. So there are two main routes into the scientific team, so the clinical scientist posts. So you can be in a trainee clinical scientist post, either via the scientist training program, so the STP, which is the national route, or through the pre-registration clinical scientist route. So there are a couple of ways to get onto the second route. One of them is through the STP equivalence, which is by the Academy of Healthcare Sciences, and but you can also have the route to registration, which is by the Association of Clinical Scientists, the ACS. And the main difference here is that the scientist training program is a national program. It is associated with a master's degree that you are doing part-time for 3 years, while the alternative route, or the equivalence route, if we can call it that, you're mostly working in the lab and you're getting some internal training. So you're not part of a national training program, but you're getting some internal training. Both of these routes could potentially take the same amount of time, 3 years. Maybe the pre-registration clinical science route could take slightly longer, but they both lead to the same outcome, which is a professional registration with a HCPC as a clinical scientist. So the outcome is the same, it's just a different route to get there. But most people are only aware of the SCP, so I think it's very important to highlight the alternative routes as well. Once you're registered as a clinical scientist, as a band 7, you're doing the clinical scientist role, and then you can move on to more senior roles. So band 8 roles, which could be like a deputy operations lead, which is basically a more senior scientist role, but has maybe a bit more managerial responsibilities as well, and then an operations lead, which again is a senior scientist with a lot more managerial role responsibilities in their job description. And again, you can go all the way up to, you know, band 9 potentially. And also, just to mention, in the previous role, the technical team, you've also got the service delivery manager roles, which are not hands-on in the lab, but they're purely managerial, but they oversee the technical side of things, and at least that's the case in the Southeast. So there's a green arrow here highlighting that it's a bit of a crossroads at the point where somebody who might be going through the technical team route reaches a band 5 or band 6 post, probably band 5, and they might have a decision to make. Do I want to continue through the technical team route, or do I want to jump into the scientist training route? And that's usually a band 5 level where people have to decide that, because they might not be as excited anymore, as committed to a technical team role, and they want to try the scientific team role. However, what we're trying to avoid is people who really want to be in the lab, going to the scientist route just because there's no career progression in the technical team. And that's why I highlighted earlier that we are actively working on, you know, new posts becoming available, new types of roles within the technical team to capture those people, so they don't have to move into the scientist team just because of a, you know, lack of career progression. But a lot of the time we've got people who do jump into the scientific team, so they might use the technical team roles as a stepping stone to an extent, but that's fine. That's part of the game, I guess. So that's what happens. And this is, roughly speaking, the two sort of career pathways that are running in parallel for the technical and the scientific team. And I think I've got another slide, Layla. Yep. So this is just a slide to show you what we've been putting in place recently in the Southeast. This is specifically due to the large number of requests that we get for work experience within genomics. So we get requests for work experience from all types of backgrounds of people. So people who are just finishing school, people who are just finishing university, people who work in a lab but not in genomics, but they want to get into genomics. So what we've put in place recently is a three-step or three-level approach. So the first level is completion of the Springpod platform, which I presume most of you, at least, who are watching this will have completed. So within the Springpod platform, there is a genomics section to it. So upon completion of that, you can get a certificate. The next step is to send that certificate to us via a specific barcode, which you can see down there, and sign up for our virtual work experience session, which is step 2. The dates shown here have passed now, but we will be releasing new dates. So this was the first time we took this approach, and we've had very good feedback, and we really enjoyed having people on these sessions. So the step 2 was capped at 30 people, and following that, so what happened during that step 2 is that we had some mini lectures, but then we also had very hands-on experience of analyzing data and reporting data. So basically what you would be doing in a laboratory, especially as a clinical scientist. So people who complete that can then move on to the step 3. So step 3 is an in-person session. Again, upon completion of step 2, you get your certificate, you sign up for step 3, and that's in person in our labs at Guy's Hospital in London. We sort of limited that to about 15 people this time around, but that includes things like lab tours, career talks. You also do a lot more interactive and hands-on experience with tasks that clinical scientists would be doing on a daily basis. So different types of tasks, but very useful, especially when it comes to you trying to show some type of work experience when you want to apply for the STP program or for any role, to be honest, in a genomics laboratory. So we will be releasing new dates for these steps 2 and 3 shortly, and this will be on the Southeast Genomic Medicine Service website. And we'll also share with COEMI as well. So that is roughly our current offering for work experience, but there's also a really good NHS guide which gives you really good pointers as to how to approach work experience and how to achieve that. So what we've included is separate to this, but this is also a really nice approach to what you could be looking at. And yeah, I mean, just to conclude, genomics is a really, really exciting field. It's developing constantly, and you know, it's at the forefront of the NHS services at the moment, and especially in the next 3, 5, 10 years, it will be part of a very large proportion of all the different investigations that are taking place within the NHS, scientifically speaking. So definitely recommend a career in genomics. And yeah, I'll hand back to you, Layla. I don't know if you want to add anything, but
Yeah, I don't know. I always say to people that if you want your job to stay the same year on year, definitely don't go into genomics. If you like consistency in every and you know you don't like change and you don't like having to adapt constantly, it's probably the worst area. But if you enjoy new developments, changes, enjoy sort of adapting to new kind of processes, it's great. It keeps you on your toes, that's for sure. But I think, is it time for questions if anyone's got anything?
Well, thank you, Nick and Layla, for such a wonderful presentation. Please, if you have any questions, pop them in the chat. While you're thinking, I might just start off with one of my own if you have any questions. So what do you enjoy most about your work in genomics?
Shall I start, Nick, and I hand over to you? I think, what do I like most? I think everything that I've kind of touched on. So I've always enjoyed puzzles from when I was a child. I enjoy trying to find the answer to things, and I still do. I do a lot of puzzles trying to figure out like escape rooms and murder mysteries and what could be the cause. And I think with genomics, I like that kind of comes into reality, that you look at a result and it's very rarely clear cut. There's usually some sort of an extra investigation you need to do, whether that's an extra lab test or whether that's emailing a different department to ask them, have they seen this before? And if they have, how did they deal with it? So for me, that part of it, I think brings through the sort of things that I always enjoyed doing, but then you get paid for it, which is good fun. Nick, do you have any?
Yeah, I definitely agree with Layla. And in addition to that, it's for me when when you do the data analysis, you do the reporting, you get an instant feeling of you contributing to something, and what you're contributing towards is somebody's health. So a patient, you're helping out a patient, you're informing them of risks for their family, for specific disorders. You helping manage the specific treatment that they might benefit from or that they should be avoiding. So it's an immediate impact, the work that you're doing on a daily basis, as opposed to other jobs where, for example, I used to be in academia, but I got to a point where even though I really enjoyed research, I couldn't see any immediate impacts from my work onto patients. So when I moved into this type of work, it was just instant. You produce something one day and the next day this has had a major impact on somebody's life. So that, in addition to what Layla said about, it's really exciting. It's always evolving as a science, and even when you do things that are considered routine tasks, they're so different to each other, each individual case. So it's always very exciting to be working in that field.
Lovely. That was such great answers. I guess, that's the beauty of science, is that it's always ever changing and you always get different things. We've got a question in the chat. What kinds of degrees would you recommend? And also, are there any books that someone could read to help generate curiosity in your field?
I'm going to have to come back on the book. There's one that I recently read that was really, really good, but I'll take on the degrees bit. I mean, there's not just a single degree. Obviously, if you're really interested in genomics, you can do a genomics degree. There's universities that offer human genomics, so you can go, if you're really interested just human genomics, you can go straight into that. Mine was just regular genomics, so I did plant animals because I wasn't sure what I wanted to go into. People do biological sciences, biomedical sciences, microbiology, all of these. In fact, I'd say most biology related degrees at the moment, the interaction I have with undergraduate departments anyway, they've got some level of genomics involved in that because of it being at the forefront of healthcare. But Nick, I actually don't know what your university degree was in.
Yeah, mine was more related to biotechnology. So you could be doing degrees in biotechnology, you could be doing pure biology degrees You know, if they've got some genetics elements in them, modules, that's even better. But, you know, you get a lot of people from biomedical science degrees as well. So there's not just one, it's not like a genetics degree that you have to do. Those exist as well, but you know, as long as it's in the sphere of biology, I guess, and science, that's what you should be looking at. And the most important thing is that it's something that you think you will really enjoy doing. You know, that's how you should be picking it, really. Regards to books, I can't think of any. You know, when I was doing my undergrad, that I was, you know, the one that was recommended to me was The Selfish Gene. That's one of the classic genetics books. But there's so many resources at the moment, so many online courses. You know, there's a genomics education program where you can find loads of resources, and I definitely recommend you look at that. For it's called GEP, Genomics Education Program. So there's a lot of information there, and then there are other courses like on Future Learn, specifically for genomics. So there's a wide variety online as well.
I've got my genomics book that I'd like to recommend. So there's one called The Genetic Age, Our Perilous Quest to Edit Life by Matthew Cobb. I really enjoyed that. I actually read it quite recently after I'd gotten into genomics, and it kind of reignited my interest. And there's also another one, can't remember off the top of my head, but it's based on Rosalind Franklin's life. So those of you that know Rosalind Franklin and the discovery of the structure of DNA know that she faced all sorts of hindrances in her life to get recognition for her work. And it's around her life and how historically women have found it difficult to progress in science and be recognized. And I think it's important to still be aware of that to this day, and make sure that it's an inclusive area. So those are two books I'd recommend.
Lovely, thank you. I'll have a look at those book recommendations myself. It's always after a new book. Do you guys have any other questions? Just give them another minute or so. But in the meantime, I'll say thank you to you both. Such a wonderful presentation, great answers to questions. I'm sure you would have inspired a new generation of scientists. So hopefully we'll see them coming along through in the near future.
Perfect.
Great, no
Thank
problem.
you very
Thanks
much.
for having us. Been really, really fun to be here and to talk to you guys. Yeah, like Komi said, hopefully we'll see you guys in the labs or in the offices soon working in genomics if this has inspired you.
Thank you.